First one, we did a crude screening, and the two extracts I'm talking about are NL, as you could see, and Odo over there, right? Those are the two extracts. We coded their names for that purpose. And we could see that NL, so the orange, excuse me if I'm color blind, but that color on your far right, yeah, it's the virus only, the untreated. The virus only there is the untreated one. So that one has 100% viral replication. And you could see our NL extract at the first concentration inhibits it more than 50%. The Odo, even much more significant, more than 90% inhibition. So that means the answer to does it inhibit HIV or not is yes, it inhibits. And the IC50s there that you see is the concentration that you need to inhibit 50% of the cells. And if it's small, it means your compound is highly effective. And you could see there we have them in nanograms and micrograms, meaning they are very small to inhibit HIV. And if you pick just a small dose, they can inhibit HIV at a higher degree. The next is, we wanted to check, okay, they inhibit HIV replication, but do they have any effect on the ones that are hiding? I hope you remember that's the challenge, right? Yes, so we wanted to use a model. There's a model in the lab that we use that has already the integrated proviruses. So we add some agents to the cells to wake the cell up. And if you wake a cell up, the virus will wake up. So if you have any agents that prevents the virus from waking up, then you can tell using that methodology. So we add the agent with or without our extract. So you could see the two agents that we use are SAHA and TNF. If you could see on the graph, the green ones, those are the agents that we use. So SAHA plus our extract NL, no reactivation occurred. That means the viruses didn't wake up. And you could see that the GFP expression has to do with the fact that the proteins that are produced from it. You could see that the proteins that are produced are very minimal for both TNF and SAHA when you add them in tandem with our extract. What it means is our extract has effect on the ones that are hiding. If any person is exposed to anything, the drugs will inhibit those viruses from becoming effective or functional. That's what it simply means here. The next is, so this is the same thing. The next is, yes, these things inhibit the viruses in not coming out and everything like that, but are they going to work if we take the viruses from the patient? Because remember, all the things I've said so far is viruses engineered in the lab. If we take virus from the patient, it's going to be the same. And you could see over there, the in vitros, the two in vitros, NL and Odo, as compared to the VO, the virus only, inhibited more than 90% of the clinical isolate, meaning the viruses from the patient. The controls, they didn't work, and that should tell you that it is very important for them to adhere to treatment because the controls there suggested the patient was conferring resistance against the known drugs. The known drugs are the AZT, route, darunavir, and virus only, coded AZT, route, and DAR. Now, the ex vivo, we took cells from a negative donor, and then we did an inhibition assay to assess whether it would be the same. And you could see there, it was the same. They inhibit HIV replication to a higher degree. The next thing is, okay, it is inhibiting replication, but which of the life cycle is it inhibiting? And we got to realize that one of the extracts called NL prevents the viruses from having their mRNA spliced to become matured. What I simply mean by that is they prevent the viruses from becoming mature viruses, and that's the NL. Odo didn't do that, but the next thing we realized Odo was doing that. Odo prevents the viruses from being integrated into the cells. So AZT you are seeing there is a known drug on the market. It's called zidovudin. And you could see our Odo is comparable to that, meaning it prevents the virus from integrating to the host genome. And that's a very key thing. Now, the last thing we wanted to tell you is, yes, these things are good in inhibiting HIV, but if someone comes to you that this drug is good, the first question you should ask them is, what is the selectivity index? What that simply means is, how selective is the compound to fighting against the virus and not our cells? Because if it's fighting against our cells, it's going to be toxic. So literature is saying if you have your selectivity index, which is SI, if you have it above 10, it's highly optimized. Below 10, be careful. Below 1, don't use it at all. And look at ours. Odo had 286.79, high above 10. NL was even in the thousands, meaning they are very selective in killing the viruses, and therefore they are safe for us to use. So in conclusion, NL and Odo, they both showed high or significant anti-HIV properties, offering a promising avenue for HIV treatment. NL extract also inhibited, as we saw, HIV splicing, preventing the virus from becoming matured. and odo inhibits general transcription of the viruses. Both extracts also had high selectivity index. Like I told you, that means they are very specific. And the next step is we confirm these findings in macrophages, which are tissues, and also do some further mechanistic studies. And the goal is to position these two drugs on the, on the scale of, on the market for functional therapy in HIV treatment. So these are my references, and I would like to acknowledge Prof. Che, my supervisors, Prof. Che, Dr. Evelyn Yaboni, Prof. Kwashi, Dr. James Abuaje, Dr. Elena Lamte, Dr. Chris Abana, and the entire team at the Noguchi Memorial Institute for Medical Research. Thank you very much.
Okay, thank you, Prince. Another round of applause for Prince and his team for this innovative, research, because it's not usual that we talk about, herbal medicine and treatment for HIV. So I think it's a new corridor for HIV cure in the future. And as you said, there are investigations ongoing for the future to see how can we have the benefits of natural treatment for HIV. So we will ask the audience now if they have questions for Prince, 2 or 3 questions, because we are limited by time. Any questions from the audience, please? Okay.
Thank you so much, Doc. Can you please clarify again the viruses that are hiding? If I have to explain to the average Ghanaian, how would you say it? Not scientific language.
Scientific. Okay, yes, please. Thank you.
Thank you.
So when the virus, infects someone, what happens is that there are some cells in your body that are not actively growing, right? They usually try and stabilize the number of cells you have in your body so that you don't overgrow, right? Now, if the viruses goes to enter into those cells, what it means is they are going to be also dormant like that. And for that reason, you are not going to show symptoms if they are in that state, right? The only way you show symptoms is if they infect cells that are actively multiplying, right? So the challenge to HIV cure is the ones that are hiding. And for that reason, if you can target those ones and eliminate them, we have a cure for HIV. Does that make sense? Thank you.
Any other question from the audience, please? Okay.
Thank you. My name is Dr. Felicia. I'm from Ghana. I want to know how far this research has gone. Is it still at the laboratory stage, or you've moved to either preclinical or the clinical stage? I want to know how far it has gone and then moving forward. So, thank you.
Yes, please. Thank you very much. So, in our last discussion with our supervisor, which were listed, they are suggesting now we go into mouse models. So we are going to do the same experiment this time around in animal models to ascertain whether they will have similar effects. And if they have there, we move on to clinicals. Yes, please. Please, is that answered? Thank you.
So thank you, Prince. If you don't have any further questions, we'll move to the next presentation about the Nigerian experience. Okay, so we have the privilege to have our colleague, Dr. Akbevwe Emanuela Besson. She is a doctor in pharmacology, graduate with over 5 years of experience in public health advocacy. She is doing also research and engagement with the community, and she focuses mainly on infectious disease. So as application, particularly in HIV studies, she's very involved, and her work includes malaria as well, and cholera vaccine advocacy, HIV prevention, and antimicrobial resistance awareness. Dr. Benson is expanding her focus to chronic and non-communicable disease as well. So today, She's going to talk about the benefits of vitamin A, tocopherol, to mitigate symptoms of azoniazid-induced neuropathy in rats. So this is, again, an innovative research that will focus on HIV tuberculosis comorbidity. So over to you, Dr. Benson, to talk about, this experience about, the benefits of vitamin E to mitigate the symptoms of azoniazid-induced neuropathy for HIV tuberculosis co-infection. Over to you.
So during one of my clinical rotations as an undergraduate student, I came across a compelling case of a middle-aged young male who was living with HIV, but was on isoniazid preventive therapy for suspected tuberculosis. Now, this patient was on a stable antiretroviral regimen, but he lives in a resource-limited setting where the routine vitamin B6, that's pyridoxine, its supplementation with isoniazid, was not the standard protocol. So after about 2 months of starting the isoniazid, this patient began to feel a tingling sensation in his palm. Now imagine the fear as this tingling progresses to numbness and burning electric shock-like pain. A textbook example, excuse me please. So imagine the fear in this patient, when this pain progresses to a non-like electric shock-like pain. A textbook example of isoniazid toxicity. Now, this patient, imagine someone who is living with HIV with its own neurological complications, experiencing this iatrogenic neuropathy, which poses a significant blow to his quality of life and the treatment regimen. Now, this patient, the the clinicians diagnosed isoniazid-induced peripheral neuropathy. And the symptoms actually progressed and got better with aggressive pyridoxine, that's vitamin B6 supplementation. But this case was the motivating spark for our research. It led us to ask, beyond the routine vitamin B6, are there other agents like vitamin E that could offer even a more robust neuroprotection and improve patient outcome? What are the underlying mechanisms? And how could this help bridge the landscape, between the complexity of HIV and tuberculosis? Good afternoon, everyone. I am Dr. Akpawe Emanuela Benson, and today my presentation is focused on finding a more sustainable, safer solution to this clinical dilemma, titled Exploring Vitamin E's Neuroprotective Mechanism in Mitigating Symptoms of Isoniazid, peripheral neuropathy, in symptoms of isoniazid peripheral neuropathy in rat model. It's a focus on HIV tuberculosis comorbidity. So as we progress, we'll be going through this content. The challenge is clear. Isoniazid, a primary agent in tuberculosis therapy, is associated with neurotoxicity, including neuropathy. Now, this is particularly concerning for patients who are co-infected with HIV, who often require isoniazid therapy. Excuse me, please. Yes. So who often require concurrent TB therapy, thereby increasing their susceptibility to drug-induced toxicity. Pyridoxine, also known as vitamin B6, is a double-edged sword. Now, it is given with isoniazid to mitigate this effect of neuropathy. But then, high doses of vitamin B6 itself induces B6 neurotoxicity. Now, imagine a case where the solution is beginning to look like a problem, and this is what made us delve into exploring vitamin E. So we sought to actually investigate vitamin E's neuroprotection, and our objective were in twofold study: to look at the vitamin E, which is tocopherol, as an adjunct, for mitigating neuropathy through the KCNN1 channel. This KCNN1 channel, excuse me. All right. So this KCNN1 channel, which is known as the calcium-activated potassium channel, subfamily N1, is a channel, is a key regulator in neural excitability. So this channel, when there's a downregulation of the KCNN1 channel, there is something what we call hyperexcitability, and this can result to nerve damage and peripheral neuropathies and symptoms of peripheral neuropathies. Now, for our second objective, like I said, we are looking at offering vitamin E as an adjunct to, as an adjunct therapy to HIV co-infected individuals. Now, for animal study, we sought ethical approval from the Faculty of Pharmacy Ethics Committee, University of Benin City, Nigeria, where 30 adult male Wister rats, were obtained, after which they were acclimatized for 14 days and administered these various doses. They were actually split into 6 groups, and each group, there were different doses attached. So we had the control group, which is the deionized water group. We had our isoniazide-only group, labeled as INH, the various doses of vitamin E, that's from the lowest to the highest dose. And then we had the pyridoxine, which is vitamin B6, that offers as a standard. So after 7 days of administration, the doses were adjusted based on the new weights of the rats. So we moved on to our skin sensitivity test, which is a behavioral study to actually assess the neurotoxicity of isoniazide and the other groups. So on day 13th, the dorsal fall of the rats were shaven, and the skin were cleaned with isopropyl alcohol to avoid for disinfection. Then on the 14th day, a disinfected needle was used to gently poke the shaven area, after which, at 4 intervals, an observer who did not know the experiment looked out for signs of withdrawal, paw lifting, and squeaking. So it's worthy to note that the experiment was carried out 1 hour after administration on the 14th day. Now, we moved on to our gene expression analysis, where the animals were sacrificed via cervical dislocation, and then the brain samples were collected, and then taken for this KCNN1 gene expression. Now, moving on to our results here, we checked, the first result is looking at the effects on skin sensitivity, the treatment on skin sensitivity. So it shows us, the average number of pokes across all groups. So when there's an increased number of pokes, it simply means that there's a greater degree of numbness. That means the animal is unable to respond quickly to the pinpricks. So as we can see here, our isoniazide-only group had a significantly higher number of pokes compared to other groups. This simply means that the isoniazide itself, actually induced neuropathy, which is the numbness to pain. Then on the other hand, we could see our various doses of vitamin E. As we can see, they had significantly reduced number of pokes compared to the isoniazide-only group. So this actually shows us that the vitamin E has a potential of, giving a neuroprotection against isoniazide toxicity. Then moving on to our KCNN1 receptor, we began to ask, okay, what underlying mechanisms does this drug have exhibit the neuroprotection? So now, as I earlier explained, KCNN1 receptor itself helps to regulate neural excitability and firing. So when there's a downregulation, as seen in our isoniazide-only group, It simply means that there is a nerve damage. And if that nerve damage progresses, you are going to have more firing leading to neuropathic pain. So as you saw here, the isoniazide group significantly downregulated the KCNN1 receptor. Now, if you look at our standard vitamin pyridoxine, which is vitamin B6, you can see that there's actually an upregulation, showing why it's a standard. It offers a neuroprotection. Then our sample itself, which is vitamin E, we can see that there is a dose-dependent protection, right, particularly at the middle dose and at the high dose. So there's a significant upregulation when compared to the isoniazide group. So this simply shows that beyond even putting out there that vitamin E has a neuroprotection, there's actually an underlying molecular mechanism behind it. So while these are preclinical studies might seem promising, vitamin E actually shows a potential as an isoniazide co-administration for patients who would need to be on isoniazide therapy for maybe suspected TB or active tuberculosis. And so our recommendation is that, further studies are being looked in for vitamin E, comparing it with the standard, which is pyridoxine, and also optimizing dosing, assessing long-term efficacy. So these are my references, but before I move on, I would like to acknowledge my undergraduate thesis supervisor, Professor Ray Ozolwa, the Department of Pharmacology and Toxicology, Faculty of Pharmacy, the University of Benin, Nigeria. I would also appreciate the University of Benin for the opportunity to carry out the research, and once again, appreciating the ICASA Abstract Committee to be able to present my research here. So these are my references. Thank you very much. So I would like to welcome any questions, concerns, or recommendations you may have. Thank you.
Thank you so much, Dr. Benson, for very interesting, finding about this research, and also extend our wishes and thanks to your team as well, because it was a team effort at the University of Benin regarding this exploration about the benefits of vitamin E in protecting, the body against the neuropathic effects of isoniazid in rats. So I don't know if we have questions in the audience. Yes?
Yes.
Over there.
Hi, everyone. My name is Lilian Jerry Mbuthi, and I'm the current country focal point in Kenya, Athena Network. Now, I'm very passionate about well-versed and comprehensive research, but my question remains one key thing that centers all. If we were to share this same research, analysis, findings, and the likes, let's say to an old grandmother somewhere in Benin, how would you simplify the language, the terms, and the jargons within that scientific research? You see, when I tell my mom something about ionized, the water groups and everything, she will not understand. So how do we break down and simplify the research language in a manner that every key stakeholder that we get to work with can be able to contribute? That is one. Two, how do we break it down and contextualizing it, again, in the languages that we speak? In each and every space that I walk into, I'm always advocating for language being a political tool and technology, even in research. So how do we break it down again? What language do we speak in Edo State in Benin? Is it Yoruba, Hausa?
Sorry, I didn't get you.
Sorry, I'm asking, what kind of language do we speak in Edo State, in Nigeria?
In Nigeria, English.
English.
Yes.
What is the local language? What is the most common one? Edo?
No. We have the pidgin, which is like common among everyone.
So I'm looking forward to the day that we'll also have our research analysis in pidgin, in Wolof. Thank you.
All right. Thank you. All right. Thank you very much. I really appreciate that comment because, yes, for most scientific research, right, when we are trying to explain to an average person, right? You might want to simplify it. So what we did, or what I did, is for every time I mention a scientific word, let's say pyridoxine, I try to infuse that is also vitamin B6, because most people might not know that vitamin B6 is also pyridoxine. And then for isoniazide, I feel like that's the only name associated to isoniazide. And then I deliberately did not mention the tocopherol, which is the scientific name of vitamin E. So I believe that putting it out there in the common name, it's better than the scientific name itself. Yeah. So I don't know if I addressed the... All right, thank you.
You can stay here. Another question? No. No. Any question, comment for Dr. Benson? Okay. So if we don't have any more questions, a last round of applause for two presenters, Mr. Prince and Dr. Emanuela, about their brilliant presentations, and they will be available in the hallways if you have further comments or questions. So this is the end of the session about this abstract-driven session, and we'll wait for the next session. Thank you very much for your attention, and as I said, they are available for additional questions if you see them in the hallway about their studies and their research in their respective countries. Thank you so much.